Hello, welcome to the DNA lecture video number 4 for biology today. Here we go. This is the section that we begin the process of discussing protein synthesis. If you don't know what synthesis means, that means making protein. Protein production is what protein synthesis is. Quick little review. Where is protein made inside of a cell? It's made in these organelles called ribosomes. Those are the protein factories of the cell. Inside of your cells, the ribosomes are located outside of your nucleus. Inside of the nucleus of your cells is where the DNA is found. So, because of that, to produce a protein, there has to be some steps in the process. So, DNA in the nucleus gets converted into RNA. Okay. The RNA is the working instructions of the DNA, and then the RNA can leave the nucleus, and then proteins are produced in the ribosomes outside of the nucleus. So they're all connected. So DNA has to become RNA, and then the RNA eventually will lead to building the protein. The process of building so protein synthesis has another name. It's called gene expression. So what we talk about is how a gene expresses itself generally means is what is the protein that you're going to make as a result of this gene. Gene expression also relates to the characteristic that an organism will have as a result of that gene. Okay. And in a lot of cases, the characteristic is a protein. What do I mean by the characteristic is a protein? Well, a protein could be a structural characteristic. It also could be an enzyme. Enzymes control how things function. So your characteristics may not just be physical structural characteristics. They could be how things work in relations to the enzymes that you're producing from those proteins. All right. A gene, small section of a DNA, we've covered this in a past chapter, that codes for a character, a hereditary character. And what I just described to you, that character is probably, in most cases, in this description, is going to be a protein. All right. Two parts of protein synthesis. Step 1 is called transcription. Step 2 is called translation. Today's lecture, this lecture is going to focus on transcription. Our last lecture will focus on translation. So we are going to focus primarily on this first step, where it shows we go from DNA to RNA, and that's called transcription. Translation takes RNA and converts it into a protein. A polypeptide is a fancy word. Polypeptides become proteins. So these are peptide bonds that hold amino acids together. Polypeptide means you have many of them. So these are chains of amino acids connected together. Chains of amino acids connected together make proteins. All right. So what are proteins made of? Proteins are made out of amino acids. All right. So today's lecture, we're going to focus on transcription, not translation. Today is all about transcription. So let's begin. What is transcription? Well, this is a process. What is it a process of? It's a process of DNA, right? So this shows you this picture. This is DNA. It gets unwound. So the double helix gets broken. The hydrogen bonds are broken in the double helix. So now we have exposed bases. These are the exposed bases in the DNA. It's showing you the image is making it bigger. So these are the exposed bases. A's, T's, G's, C, T, A, G, G, C. These exposed bases then get converted into an RNA, or in this case, mRNA, messenger RNA. So the A bonds to a U, and that's how the U knows to form. T goes to A, G goes to C, C to G, T to A to U, G to C, G to C, and C to G. Okay, so this is the complementary base pairing rules for DNA to RNA, or mRNA in this case. Okay, there's no T's in RNA, so you don't see any T's in this green area down here. You and me, we have a nucleus. So where does this take place? In our cells, it takes place inside of the nucleus. Okay. Bacteria, prokaryotes do not have a nucleus. This takes place inside of their cytoplasm. Okay. So this is the process of taking DNA and eventually converting it into mRNA. All right. That's what transcription is. Why does this need to take place? Because DNA cannot leave the nucleus. mRNA can, DNA cannot. Steps of transcription. All right, so here are the steps. Here's a picture that shows you it. There's a new enzyme. We learned about DNA polymerase in a previous lecture. Now we're talking about RNA polymerase. Well, DNA polymerase makes more DNA. RNA polymerase makes new RNA. That's how it works. So what is it? How does it work? Well, RNA polymerase is an enzyme. all right? It has to bind or stick to the DNA in a region. So what region of the DNA does it bind or stick to? It's called the promoter region, all right? So RNA polymerase, how does it know where to attach to DNA and do its job? Well, it looks for a coding, an area that is coded for the promoter. RNA polymerase lands on the promoter, and then the enzyme starts moving along the DNA strand, the DNA double strand, and starts unwinding it and breaking hydrogen bonds as it moves along. So what is RNA polymerase doing? It binds to it and starts moving along and reading it. Okay, so this is step one. Step one is binding, all right? And they also call that initiation. That's the word that is used in some textbooks, okay? So in order to turn DNA into RNA or mRNA, you need RNA polymerase. Step one, it attaches to the promoter region of the DNA molecule. Step two, all right? So this is making that picture bigger. This is RNA polymerase. It moves along the DNA, and as it moves along, it breaks these hydrogen bonds holding DNA together. Excuse me. Okay, as it moves along, it adds complementary bases. Okay, so it adds complementary RNA nucleotides, not DNA nucleotides. All right. As it moves along, the previous regions reattach themselves, and then the RNA starts peeling off of it. So this is newly made RNA in pink here. Excuse me. Excuse me. Dry throat there. All right. Okay. So here we go. Next slide. All right. The final thing is RNA polymerase gets to the end. The end is called the terminal sequence, or in some cases they call it the terminator, or in termination. Same thing. So it gets to a sequence of DNA that says, hey, you're done. So unattach, release the mRNA, and unattach yourself. And that's what happens at the terminal signal, okay? RNA polymerase releases, the new mRNA or RNA is released, and now this could leave the nucleus, all right? What kind of RNA is made this way? All of them are made the same way. Excuse me, allergies are hitting me during the video, all right? So let's take a review of the end sequence, the end of it. So the end of it is it hits the terminal sequence. It releases. The RNA is released. What is the RNA? The RNA could be any type. mRNA is made this way. tRNA is made this way. rRNA is made this way. All RNA is made the same way. And this is just showing you transcription again. You start with DNA and you make RNA. Okay. T becomes an A becomes a U, C becomes a G, T becomes an A becomes a U, G becomes a C, and so on and so forth. Now, in our next lecture, we're going to talk about the next part called translation. Translation is where this RNA sequence here that's produced gets converted into a protein, or in this case, called a polypeptide. All right, we will stop there.